Hi, everyone. I'm Linda Tintawi, CEO of the Lustgarten Foundation. Welcome to our Lustgarten Live 2023 Year in Review. And what a remarkable 25th anniversary year it's been. Lustgarten-funded science has been a driving force in every major development in pancreatic cancer research since our founding in 1998. As the world's largest private funder of pancreatic cancer research, we aim to advance breakthroughs in the understanding of the initiation and progression of pancreatic cancer to improve the diagnosis and treatment of the disease and provide more hope for the future. We fund high-risk, high-reward research focused on early detection and interception, therapeutic development, and personalized medicine. We are at an inflection point where we're seeing advances in pancreatic cancer research move at a faster pace more than ever. Our efforts at the Lustgarten Foundation are guided by our Chief Scientist, Dr. David Tuveson, and Chief Medical Advisor, Dr. Elizabeth Jaffe, and our Vice President of Research, Dr. Andrew Raikman. Thank you. I hope you will enjoy this informative discussion recorded earlier this month.
So one of the most exciting advances in pancreas cancer research, and one of the things we've seen some clinical traction in this year, has been the emergence of drugs that can inhibit KRAS, or the smoking gun of pancreas cancer. So Dr. Tuveson, could you tell us a little bit about what we've seen coming out of these clinical trials and why this is such an exciting advance for pancreas cancer?
Thank you, Dr. Raikman. KRAS is the gene which is mutated the most commonly in pancreatic cancer. It's also the cause of initiating the disease and keeping it going as it progresses. This gene makes a protein which acts differently than a normal KRAS protein. It acts in such a fashion that it drives the cell to proliferate more and resist medicines. We've thought for a long time that any drug that could make the bad protein go away may be useful for patients, because we knew in scientific experiments in the laboratory that when we got rid of the gene, pancreas cancer collapsed. Fortunately, over the past 10 years, chemicals have been made that target mutated KRAS protein. Most recently, some chemicals became available which target the one found commonly in pancreas cancer. These are mutations in the early part of the gene. In the laboratory, these drugs look good, but really, we don't know if they work at all until patients receive them. And over the last year and a half, around several dozen patients have received these new KRAS drugs. There's one made by Revolution Medicine, and we recently got the information back from physicians treating those patients at a clinical conference in Europe called ESMO. The trial, which is a phase 1 trial, which again are designed to ensure that whatever medical treatment is not dangerous for a patient, and to try to work initially on how that drug may work in patients. This trial showed impressively that the majority of patients who received this medicine had stabilization of their disease, and a fraction of them also had shrinkage of their disease. These are patients with advanced pancreatic cancer, and to get this response in just a few weeks and a few months of treatment is very impressive. Also, the side effects from this medicine appear to be very tolerated. And because they're tolerable, that's great, because now we can combine them with other medicines. And so this is actually our first glimpse at KRAS drugs. We're quite excited about it. It shows that decades of research in the laboratory to identify the driving genes in pancreas cancer can lead academics and the biopharma industry to create molecules which can target those dependencies. So this is actually a huge moment for pancreas cancer. I have discussed these results with Dr. Jaffe and Dr. Knox from Toronto, and we think this could become a backbone for how we treat patients as we potentially add on new therapies, such as the ones that you'll hear about from Dr. Jaffe today. So what we would like to do is push this with our partners in academia and the biopharma industry to see how good are these drugs? How long can we treat a pancreas cancer patient with these medicines? What are the things we can combine with them to ensure that they get the biggest effect for the longest period of time? What we've learned from other cancers so far is when you have one drug, it can look really great, but oftentimes it's not for very long. And you need to combine with other medicines for that reason. And so this is, I would say, the breakthrough that I've been waiting for as really a scientist and a physician in this disease, because now I think we will have a weapon to make the cancer less tough. And now a lot of other ideas, I think, will have a better chance of working.
So 2023 also saw some wonderful advances in the field of immunotherapies for pancreas cancer. And that's an area that's been very challenging to bring forward for patients with pancreatic cancer. But Dr. Jaffe, something you've really dedicated your career to and really been steadfast in the belief that these were going to be transformative. So it's particularly exciting to see the kinds of responses we've seen in patients with some more advanced combinations, including vaccines and immunotherapies in people at different stages with pancreatic cancer. So I'm hoping you could tell us a little bit about what's come out this past year and why that's so important for patients with pancreatic cancer.
Yes, thank you, Andy. I've been working on trying to activate the patient's own immune system to fight cancer for 30 years. It's been a dream of mine that we would be able to use the patient's own immune system that if done correctly, can see changes in the cancer that are different from the normal tissue that the cancer derived from, in this case, from pancreas cells to pancreatic cancer, thereby getting rid of the cancer, but not causing the toxicities that we often see with other therapies such as chemotherapy and radiation therapy. And it really was an aspiration, a dream, until probably about 10 years ago, when something happened. We had a breakthrough in the field of immunotherapy. We have these agents called immune checkpoint inhibitors. They're antibodies that can actually be able to further activate an immune cell to fight cancer by turning off what we call a stop signal. So immune cells can see cancer, but they're often stopped. And pancreatic cancer is able to do that in many ways. It has many different signals that tell the immune cell, go away, do not come and try to kill me. However, these immune checkpoint agents, which are antibodies, can turn off these stop signals and allow the immune cells to actually see cancer. This has worked very well with just one antibody in cancers like lung cancer and melanoma. It's still been a great challenge in pancreatic cancer. So the field in pancreatic cancer has really tried to better understand, are there additional signals we have to try to turn off or turn on, or is it that the immune cells aren't there yet? Well, one of the problems is in pancreatic cancer, unfortunately, the cancer is so insidious, it hides itself so well from the immune system that we never really make these immune cells. Well, one of my colleagues, a group who led a group at Memorial Sloan Kettering, Dr. Balachandran, was able to think about this from a different way. He said, if we can now sequence the tumor and see what these new proteins are that the pancreas tumor has that the normal cell doesn't, can we make a vaccine that can actually induce or activate these immune cells so that they can see the cancer? And so giving this vaccine to a small group of patients after they had their tumor removed at surgery, he was able to show that he can get longer-term survival. And the immune system was seeing these proteins that he put into the vaccine. Now, he had to give it with one of the antibodies available that can turn off that stop signal, because once you make a good T cell, they're still going to be they're still going to turn on these stop signals once they get into the tumor. So this combination of a vaccine plus an antibody called anti-PD-1, you may have heard Keytruda, there's other names for it, giving that together with the vaccine was able in a small group of patients to have longer-term survival. Well, this is exciting because he is now working with a company, Roche, and Genentech, they're one company, to look at this vaccine in a larger group of patients at multiple different cancer centers. So we'll see, we're very excited. Now, work that I've been doing for many years is trying to understand what are those other signals that we also have to either turn off or even turn on, because immune cells are regulated. So they have stop signals and they have activating signals. Maybe we need to do both to get the most potent immune cell to fight pancreatic cancer. It's a tough cancer. So we've been doing what we call a platform study, where we're able to test our immunotherapy before a patient goes to surgery. So we can get a pre-treatment biopsy, and then we can get the tumor at the time of surgery and see how our treatment has worked on the tumor. This is very important because with all our new technologies, we're able to really understand at a molecular level. So the different proteins within one cell, we can understand how they're changing with our therapy. We can look at all the cells in the tumor and in a short period of time, understand many of the signals that are changing with our therapy. And so by doing this, we can build on what we learned from each trial. So the first trial was just with the vaccine alone that we had developed, and we saw some good changes. We learned that we needed to add a second immunotherapy. We learned a lot from that as well. We learned that we needed to add a third immunotherapy. So now this triple therapy approach, it's a vaccine, it's an antibody that turns off the stop signal, and it's an antibody that accelerates. Okay, so it further activates the immune cells. And these three together, we're now seeing in a small study, we're seeing evidence that the tumors are actually responding to this therapy. And in a handful of our patients, we're actually seeing a near complete response after one triple dose. And we believe that this could mean that we'll get longer term survival. So really, to summarize, I think science in patients, which is something that the Lustgarten Foundation has been funding for many years now, is finally yielding important information, and it's actually having benefit in our patients. And we're hoping in the next few years to have a good triple or even 4-agent combination that activates the immune system so completely that patients will have very long survival.
As many people know, one of the areas that's been a huge issue for pancreas cancer is that we often diagnose it too late to have the kind of potentially curative therapies that we know work when we catch people at earlier stages. So it's been a huge effort for the foundation and for the field overall to try to find ways to detect people earlier in disease. And one of the challenges there is we don't often understand who is at risk. And there's been some advances this year and over the past couple of years in really being able to demonstrate that if we find people at risk, we can find pancreatic cancers earlier and people do better long term. And being able to use over this past year reports using new technologies like machine learning to dig deeper into health records to find people at risk who we wouldn't have found through the kinds of things that we typically look at, such as family history or environmental exposure. So I think that this is something that's really exciting, and it's setting us up for a new era of earlier detection and potentially being able to intervene even earlier in disease. So Dr. Tubeson, hopefully you can give us some more insights into why this is such an important problem and why this is giving hope for the future.
Thank you, Dr. Rakeman. Pancreas cancer is a disease that's gotten more common over the last several decades. It now strikes about 1 out of 50 people throughout their lifetime. And trying to identify individuals before they get pancreas cancer, of course, been one of the most important goals of the field and of the Lustgarten Foundation. Now, we have supported work at Johns Hopkins Hospital for a number of years, and including the work of Dr. Jaffe that you just have heard about. And the work, though, that I'm talking about was really initiated by Dr. Ralph Ruban, the head of pathology, the director of that department at Johns Hopkins. He assembled the first cohort of families with pancreas cancer to try to understand the genes underlying these families. And over the last several decades, many of the genes that predispose for developing pancreas cancer have been identified in this fashion. Because families have been identified which are at increased risk for developing pancreas cancer, Johns Hopkins was able to start a surveillance program for high-risk patients called the CAPS program. And the CAPS program was led by Michael Goggins, a gastroenterologist at Johns Hopkins, and Mimi Canto. They published recently through surveillance studies where you use endoscopic ultrasound and MRI and blood tests to monitor individuals, you could pick up early pancreas cancer in patients who are at high risk. And the folks they picked up early, they had great outcomes. They received surgery and they had small tumors resected. And 5 to 10 years later, they still don't have cancer. And so this is exactly the outcome we've been looking for a long time, that early detection can save lives. Just like when you get colonoscopy to find a small polyp in your intestine, same concept. Now, how do you apply this logic to the overall population? About 10% of patients have that family history. 90% don't. So how do you help the most individuals who are at risk of pancreas cancer, about 1 out of 50 people, as I said? Well, fortunately, we've been able to engage some really brilliant scientists and physicians to help us work on this. One is Dr. Chris Saunders, who's a mathematician and systems biologist at Dana-Farber Cancer Institute in Boston. And Dr. Saunders conducted a study where he worked with the Danish government and with the VA health system. And so the Danish government, country of Denmark, they have a socialized medical system where they have all the medical records on all the people who live there. Sounds simple, but in our own country, it's hard to do that because we have different health systems and hospitals with different opinions about how to take care of people. And Dr. Saunders was able to get all the information over many years in Denmark of how people lived in health and what made them sick. And by looking in the medical records of hundreds of thousands and then millions of people, he could look backwards in time to the folks who ended up with pancreas cancer, comparing them to individuals who are otherwise similar who didn't get pancreas cancer. Using computational approaches and machine learning methods, he was able to actually see patterns in their medical records that us scientists and doctors had not even thought about before. It turns out that before you are diagnosed with pancreas cancer, you end up going to the doctor a lot more often than you have in the past for all kinds of reasons. Your medicines are changing, you have more infections than usual, you have lots of things that bring you to the doctor that didn't in the past. Sometimes a stomachache, sometimes reflux or indigestion. But sometimes it's just, you've started to lose a bit of weight, you go to the bathroom more at night, all kinds of things that are just could be caused by almost anything. But he was able to apply computational power to come up with things that occur in the lives of normal people that would be warning signs if we took more comprehensive care of people. So this is Denmark. Whenever you find something, you have to go somewhere else to see, are you right? You cross-validate. And so Chris came to America and looked in the VA medical records, which is our closest thing to a socialized medical system in our own country. And he had to work a little bit harder because the system was set up differently and not all the records were present. And he had to, normalize things just like a scientist would do when they do their own study. But he was able to find the same patterns here in America. So this was actually very exciting. And this was published very recently and has really set the stage for delving more deeply into this, applying data science, computational methods. We call it artificial intelligence in the media, but now you think that's a dangerous thing. So I'll use another term here. But the bottom line is, could we learn more about this disease by just paying more attention to big populations of people? And I think we can. The reason why this is so important, what Chris found, is he works with Brian Wolpin, a very distinguished medical oncologist at Dana-Farber Cancer Institute who runs their GI oncology practice. Brian, for his career, has been trying to figure out a way to stratify people into high risk, medium risk, and low risk for the concern about will they get pancreas cancer or not in the future. Brian has known for some time that the high risk patients, the ones that have genes that they've been inherited because they have a family history of pancreas cancer, need to be followed in the way that Mike Goggins and Johns Hopkins figured out, the CAPS program, surveillance scanning and blood tests and office visits. But what about the intermediate risk and the low risk? Well, the information from Chris's computational approach helps flesh out what Brian needs to tell someone they're either low risk or intermediate risk. And the beauty of that is then the chances that studying them will be beneficial to that patient have gone way up. And so Brian works with Chris, and they both have had, really terrific advances over this past year, thinking about ways to use today's information to inform doctors that individuals may be getting pancreas cancer. That's not a new test. It's just paying more attention to the information that we already have. The new test thing is obviously very important. What do I mean by that? Well, I said colonoscopy for colon cancer. Just like years ago, we developed Pap smears for cervical cancer. What do we have for pancreas cancer? Well, I don't think there's going to be a colonoscopy or a Pap smear for pancreas cancer. There's going to be, though, better imaging methods, and better imaging methods are coming. We have invested in that also with Johns Hopkins, and there's a program called Project Felix, which has that. And that was something that was mentioned recently when we had a special meeting, a special symposium. And there are going to be newer ways, newer molecular imaging ways that we're going to be developing over these next few months and few years that we're very excited about. Now, the reason I'm excited about early detection is I'm sure we're going to figure out a better way to do it. But again, it's 1 out of 50 people, and actually that get pancreas cancer, but we're all at risk for pancreas cancer. We all have the earliest signs, meaning the pre-cancers. And my colleague, Dr. Jaffe, actually knows that, and she's trying to fix that. And I would like to introduce her back to you so that she could tell us how she's taking on the harder problem, which is trying to reduce that risk across the population by studying people that she knows has the earliest pre-cancers.
Thank you, Dave. Yes, it's a real clinical problem that we are faced with. And one, not knowing who to take to surgery, because that's our only treatment to prevent the pre-cancer from becoming a cancer. And two, when we do take them to surgery, we're taking out a good part of their pancreas. So a patient really has a lot of problems after the surgery, and their life quality is somewhat altered due to losing a good part of their pancreas. So the goal would be to try to preserve the normal pancreas and just eradicate the pre-cancers as they develop. And so thanks to Dave, actually, who developed this amazing mouse model of early pancreatic cancer development, we now know that mutated KRAS, along with some other genes that occur, some other genes that are activated later on, help to move a normal cell into a precancer and then into a cancer. And mutated KRAS, as we've heard, is usually the first mutation that can be detected in the precancer. And that's at a time when the immune system is on high alert. However, if we allow the precancer to continue to progress, the immune system gets turned off. And we know this from the animal model that Dave developed. We've published work on this, and we've also been able to develop a vaccine similar to the human papillomavirus vaccine that prevents cervical cancer and other cancers associated with that virus. We've developed a vaccine that targets mutated KRAS. And this vaccine, when given very early, can activate a KRAS-specific immune response that appears to halt, at least in the mouse, progression to cancer long-term. And so we're now testing this in the high-risk population that Dave described to you to see if we can actually activate an immune response, and then also in patients who do need to go to surgery to get rid of their precancer, we can take a look and see if the immune response we've activated can halt that progression, even eradicate it. So it's still early days, but we're very excited that we finally have not only the knowledge, but also the tools to begin to try to prevent pancreatic cancer from progressing in these patients who are high risk. Long term, the dream would be to vaccinate all people, because these vaccines are quite safe to prevent pancreatic cancer in the future. Again, this is a long-term goal, but we're getting started. And thanks to the Lustgarten Foundation, we have the funding support to push this work forward.
So while we're all excited for a future where we can have targeted therapies and immunotherapies that will take care of pancreatic cancer, we know that for, at least for the time being, chemotherapies are an important tool in our toolbox to use for patients, but there's often some frustration about not really knowing what's the right chemotherapy to choose and what's the right way to use it in individual patients. But there's been some traction and some progress this year, thinking about how to use chemotherapies differently and how to match the right therapy with the right patient. But Dr. Jaffe, even more so, I know that there's work that you're engaged with that's really looking to upend how we think about pancreas cancer and how we think about heterogeneity of the disease or differences between individual patients and how to match and personalize even the chemotherapies that we use for patients. So I'm hoping you could talk a little bit about some of these clinical advances that have happened with using chemotherapy and how to select the right chemotherapy, but also point us to what to look forward in the future coming out and what we're going to learn about how to individualize therapy for patients with pancreatic cancer.
Yes, thanks, Andy. So chemotherapy has been probably the best advance we've had so far for metastatic pancreatic cancer. And it started when gemcitabine was identified, and then Abraxane was added. So the gemcitabine Abraxane was a major advance for these patients, giving a progression-free survival of about 5 or 6 months and overall survival of 11 to 12 months, which seems quite good compared to what we had before, but certainly not where we want to be for patients with advanced pancreatic cancer. After that, a second combination chemotherapy called FOLFIRINOX was developed, and that seemed to give a little bit more benefit in the studies that were done, and there were a number of large studies in different patient populations. And FOLFIRINOX may give a 1 or 2 month benefit in progression-free survival and overall survival, but it is more toxic. And since then, we've had an even smaller, but still advance that has occurred in the past year. And this is with a regimen that makes a little change on FOLFIRINOX, and it's called NALIRIFOX, sorry. And what's different about that is that it actually takes one of the agents in FOLFIRINOX called irinotecan and puts it into a liposome, which makes it a little less toxic and a little bit more effective, similar to what happened with Abraxane, which is again an agent, a chemotherapy that's in a liposome. So the progression-free survival is maybe 1 month better than FOLFIRINOX. The overall survival is still about 11 months, maybe a little less toxic. So a small win. But the real issue with chemotherapy is we don't know who's going to respond to which of these 3 chemotherapy agents, combination agents. So will a patient respond to Gem/Abraxane? Will they respond to FOLFIRINOX or to the modification of FOLFIRINOX? The problem here is that these patients don't have a lot of time to determine which of these chemotherapies is going to be best for them. They're already not feeling great. They have metastatic cancer. We all know that pancreatic cancer, when it's metastasized, can move quickly. So the Lustgarten Foundation has been supporting an innovative new study called PASS-1. And this has been a collaboration between Stand Up to Cancer, Lustgarten Foundation, and the Canadian Pancreatic Cancer Foundation. And this is an important study because in other countries, such as Canada, you only have one chance to pick the right chemotherapy because insurance won't pay for more than one. But again, when we look at the pancreatic cancer under a microscope, we can't tell which of these chemotherapies is going to work best on our patients. And so the PASS-1 study is comparing FOLFIRINOX and Gem/Abraxane to really do a deep dive into each patient's tumor before they go on the chemotherapies. We're looking at the genetics, we're looking at the epigenetics, we're looking at the proteomics, and we're making organoids where we can test different chemotherapy agents to determine which ones the patient's own cancer will respond to. And we're doing this very quickly in a short period of time while we're testing the chemotherapies, while we're treating the patients with the chemotherapies. And so the organoid data has already been informative to tell us if a patient isn't responding or if they stop responding to one of those chemotherapies, maybe they would do better with a different combination. In addition, the genetics is also giving us information that's very important to predicting who will respond to these therapies. So right now, the study is still ongoing. We expect to finish enrollment. It's a multi-center study, many centers in the US and 2 centers in Canada. We'll have the completed enrollment by probably the first quarter of 2024. And we're already beginning to assess all of the information that we're getting from these studies that we're doing on each patient's tumors. So hopefully this will yield important information, what we call biomarkers, to predict early on at the time of diagnosis of pancreatic cancer, which of these therapies would be best for these patients and what additional therapies may be important to even enhance the current chemotherapies that we have available to our patients.
This is very exciting times and really looking forward. I think this
Yeah.
PASS-1 data is going to really transform the way we think about pancreas cancer for many years to come.
I think it will, and it'll give patients a very early chance of responding to the first therapy we try, which will be very important. That is critical in these advanced cancer patients.
2023 is an important year for the Lustgarten Foundation. This marks our 25th anniversary. That means 25 years of finding the way to make pancreatic cancer a curable disease through the supportive research. And there's a lot for us to celebrate in 25 years, a lot of successes that have happened, a lot of transformation, a lot of new understandings about this disease and changes in the way we care for patients. But it also recognizes that there's a lot more to go. We haven't finished the mission yet. There's still work to do. So rather than spending the year looking backwards or celebrating on our accomplishments and resting on our laurels, we really took the approach of let's just use this as an opportunity to set the stage for what's to come and really analyze what are the challenges to come, what are the most important issues for us to address, and how are we set up because of this 25 years of supporting research to move us into that next phase. So to do that, we organized and ran a first ever public scientific symposium focused on pancreatic cancer, supported by the Lustgarten Foundation, called Seeing Beyond. It was a fantastic event, brought together top researchers to come in and talk about their work. And what we really charged them with was to talk about not just what's happened, but what's to come and what's going forward. So Dr. Tuvison, you were instrumental in organizing this. You were helped to manage the sessions. I'm hoping you could give us some of your impressions about how that symposium went and what you see as coming out as the challenges and opportunities for the future, so that we can continue to make progress and accelerate our progress going forward.
Thank you, Dr. Rakeman. Yes, we had a really, I think, terrific moment when we could gather together the thought leaders in the pancreas cancer community in middle of November in Manhattan to discuss current state of affairs in pancreas cancer research and medicine. And so our symposium really was supposed to acknowledge what we've accomplished, but more importantly, help chart the map for what we need to do to complete our tasks, so that we have much better approaches for all individuals regarding pancreas cancer. And so we invited several dozen luminaries from around the country to come and attend this symposium, and a subset of them to speak on germane topics. One of the topics was the concept of early detection and risk for developing pancreatic cancer. Again, pancreas cancer has become more common. 1 out of 50 people will get pancreatic cancer. We want to know which one out of the 50. And we want to know for the other 49, what can we do to make it so they always stay with lacking pancreatic cancer? And so we had presentations from Johns Hopkins, from Anne-Marie Lennon and Mimi Canto, which talked about several different approaches, including the CAPS study, where they're doing intensive surveillance for individuals who have a family history of pancreas cancer, and another study which is in parallel for patients who have cysts in their pancreas, where again, they're following the patients carefully using endoscopic and approaches, as well as MRIs, and biopsying the cyst fluid, et cetera. What are various ways that they have developed that they see as a path forward for such individuals? Dr. Peter Allen from Duke University, who runs the surgery department and surgical oncology there, himself studies patients with cysts, a particular one called IPMN. This is a very common condition. And he's trying to decide the patients with IPMN, which ones have benign ones versus ones that will become a malignant pancreas cancer. And he has a surveillance program as well as an intervention strategy where he's treating them with anti-inflammatory medicines to see if he can reduce the risk of progression of IPMNs in patients. Finally, Brian Wolpen from Dana-Farber, who runs the GI oncology program there, talks about how all that information helps fit into the models that he's devised on risk stratification in order to develop metrics that can be used by any doctor throughout the land to help advise patients on whether they are at risk or not of developing pancreas cancer and what are those worrisome signs that they imminently may be developing pancreas cancer, using electronic health records as well as blood-based monitoring, blood tests, as well as imaging. And so that was our first session talking about this topic, something that really affects all of us. We all, are at risk of cancer of various types. Is there a way to lower that risk for pancreas cancer across the population? So that was our first session on this, and we got a lot of questions from the audience, and I think it was a really dynamic engagement of the faculty and the audience. We then segued into the therapy space, and we had another session where we had lectures on the biology of pancreas cancer and how it impacts therapeutic response from Dr. Zhenzhen Ye, who's a head surgeon at University of North Carolina, and chemotherapy and smarter ways to use chemotherapy from Dr. Jennifer Knox, who's a head medical oncologist in the University of Toronto, Princess Margaret Hospital. And Jennifer co-directs the PASS-1 clinical trial with Dr. Jaffe. And then finally, an immunotherapy talk from Dr. Vinod Balachandran, who, as you heard from Dr. Jaffe, has devised some novel immune therapy approaches using mRNA vaccines. And they all talked about how the scientific knowledge in each of their areas has led to new clinical insights and how they're starting to see those clinical insights having positive impact on patient outcomes. That's, I guess, for me, the most exciting thing, that this big investment by the Lustgarten Foundation and all the people we've affected over the last 25 years has led to the realization that deep science will lead to great clinical results. And we're starting to see it now. So it was really that 25-year investment in understanding the disease so that we can be smarter when we go into the clinic. And we saw that really in session 2, and that's sort of our leaning forward approach. Now, we had those 2 sessions, early detection, risk stratification, better medicines, which are sort of the balancing act in pancreas cancer. And then for our finale, we brought in Professor Bert Vogelstein, who in many ways is our magician in this disease. He is, the leading geneticist at Johns Hopkins. He helped identify some of the most important cancer genes known in colon cancer, and then applied his approaches to pancreas cancer 15 years ago. Bert has been focused on pancreas cancer these past 10 years, and his insights have been very helpful. He's been able to work with many of the people I mentioned just now to help them with their clinical approaches, and even with Dr. Jaffe, who's his colleague at Johns Hopkins. Bert's interest is in using genetics to derive better approaches, either through understanding the disease better or designing better therapies. He talked about several things. He would like to see us to really rethink this issue about the genes involved in pancreas cancer. The KRAS gene, which now has a medicine that targets the protein product of the gene. He knows it's important. He wonders if there's one other genetic event. He has a theoretical framework for posing that challenge. I would say it's a topic that led to many people discussing things. There's no evidence yet of knowing what that other event might be. It might be an epigenetic event, meaning not a change in the gene, but rather a change in expression of genes. But the importance of him stating this as a challenge to our field is now other people can think differently. And maybe there is something else that we could, if we understood it, prevent it. Just like Dr. Jaffe wants to prevent it by vaccinating, maybe we could prevent an epigenetic change also with a medication change for individuals. Bert also is trying to devise better immune therapy approaches. He has, I would have to say, creative approaches for that, using synthetic T cells as well as antibodies, which are hybrids, where you have one arm binding one thing, another arm binding another thing. He has a terrific group of individuals in his laboratory who have really worked on his ideas. Bert gave a challenge talk to our field, and he wasn't meant there to pacify us or pat us on the back. He was meant there to keep us moving. And I think everyone appreciated it in that regard. He thinks we need better ways, better approaches for therapies, better detection methods, better understanding of pancreas cancer. So, you know, many people, when they reach that age and have accomplished so much, they think about what fishing pole do I buy next? Bert doesn't. He thinks about what problem do I solve next? And our field is blessed to have people like him in it. And it was this fitting tribute to the conference, which we set up. And so, again, the conference, I call it bittersweet. It's sweet in that our investment in science has led us to where it is now. It's bitter that we're still actually working on pancreas cancer. We don't want to have a 50th anniversary conference. We want to be done. And so I think that's part of our message today. Your support has led us to this point. With more support, we won't have to have a 50th year anniversary. And now back to Dr. Rakeman to talk about that.
Thanks so much, Dr. Stevenson. And it was an inspiring day to listen to everyone and listen to the progress that's happening. And it's clear that there's been lots of progress made. It's clear that we haven't done all that we need to do. But it's also clear that people are now asking the right questions. We don't have all the answers, but people are asking the right questions. And that because of support from all the people that helped make Lustgarten possible, because of the community of scientists and patients that Lustgarten generates and brings together, that this work is continuing to move forward. And certainly the time when, with the momentum that we have, it's not time to rest on our laurels, but it's time to press the accelerator harder and continue to push on this, because the tools are there, the knowledge is there. It's the hard work that needs to happen now to get it done. So one thing most people understand is that pancreas cancer is a hard problem. It's one that no one group is going to solve on their own. And as much as we need collaboration among scientists and new ideas, we also need collaborations among the funders to drive things forward, partnerships that bring in additional talents, additional resources, and additional dollars to help drive things forward. So one advance that we're very excited about in 2023 is a partnership announced between the Lustgarten Foundation and the Cancer Research Institute that we're calling Inspire, which is focused on driving immunotherapy research in pancreatic cancer going forward. And this is a partnership that's based on a longstanding history of Lustgarten Foundation supporting pancreatic cancer research, and a longstanding history of the Cancer Research Institute supporting immuno-oncology research. And I know these are two groups, Dr. Jaffe, that you've been involved with in a very deep way for a long time. And I'm hoping you could give a little bit of your perspective on why this partnership is exciting, and what kinds of additional resources and additional know-how that working with a partner like CRI can bring to pancreatic cancer research.
Yes, thanks, Andy. I've been working with the Cancer Research Institute, CRI, for many, many years. They've been around for over 50 years as an institute in New York. So not everyone has heard about them, but they've certainly been able to engage many scientists who are immunologists to work with them to take science that we've learned about immunology and test it in new therapies for patients. I've worked with them for a long time. I'm an immunologist who applies what I've learned to pancreatic cancer, with the goal, of course, of trying to use the immune system to treat and eventually prevent pancreatic cancer. They have focused on other cancers that have traditionally been more responsive to the immune system, such as malignant melanoma, skin cancer, or more recently lung cancer, which we now know responds to some of these new agents called immune checkpoint inhibitors. Well, the Cancer Research Institute has funded a lot of the early studies that have led to identifying these new immune checkpoint inhibitors that are antibodies that are able to take off the brake on immune cells and allow the immune cells to kill lung cancer or malignant melanoma. The problem is they never really concentrated on pancreatic cancer until recently. They didn't see that the immune system had a role in pancreatic cancer. And over the past, I'd say 5 years, they have finally, in my mind, seen what I've been seeing for 25 years. That is that there are ways to get the immune system to see pancreatic cancer. It's just a little bit more challenging, in fact, a lot more challenging to do that. And so they have partnered originally with the Parker Institute on an earlier study called the Revolution Study. And I actually have been part of the scientific advisory board for that study, and I serve as the chair of the Data Safety Monitoring Board of that ongoing study, which is a very interesting study, but the Parker Institute has moved in different directions. Fortunately, the Cancer Research Institute said this is a study that would be important to continue. Myself and others at the Lustgarten Foundation want to see it continue. And so this is a great partnership for this national platform study, which is trying to look at combinations of immunotherapy in patients with advanced pancreatic cancer, so new metastatic disease, and try to get the best combinations to try to treat pancreatic cancer by activating the immune system. And so I'm very happy to be a part of bringing the two institutions or two foundations together because they're both so powerful. One in this great knowledge of immunotherapy, and also the CRI has partnered with many of the pharmaceutical companies that have some of these agents. So they have a long history of that partnership. We at Lustgarten have the knowledge of pancreatic cancer, and we've funded many early studies in pancreatic cancer to better understand what combinations are needed. This is a win-win situation. I'm so excited to be working with both foundations, working together to try to really make a big impact on pancreatic cancer. And as Andy has said, this is the time to do it. We have a lot of knowledge. We have lots of interest nationally. We need the funding to continue to push our agendas forward. We are at an unprecedented time, and we can really make a difference for patients with pancreatic cancer. I'm more excited than I've ever been to be a part of both groups working together.
Thanks, Dr. Jaffe. And it's been fantastic to work with our colleagues at CRI. I think it's safe to say they come with the same level of enthusiasm and passion about the science and about the patients, most importantly, that we do at Lustgarten. And I, for one, am extremely excited to see this partnership grow and flourish and to see how much more we're going to be able to do by working together with our partners. So over this past year, Lustgarten has been able to launch a new program that's going to be extremely impactful for the way we think about clinical research, in particular in pancreatic cancer. This is a project that we're calling LUCID, which is Lustgarten's Unified Clinical Information Database, where we're seeking to bring together data from lots of different clinical trials, including the clinical trials that we support, into one place. And I know, Dr. Jaffe, this is something you've been really passionately driving for a long time, especially looking at how clinical trials have advanced so much and we're able to collect so much information from the patients that we treat. And we really want to be able to ensure that we use all of that information to its fullest extent, that, patients are our most valuable resource at the end of the day. And we really want to honor the contributions that they make to clinical trials by making sure that that data is as useful as possible. So we're in the early stages of building out this database, but I hope you could, give everyone a little bit of color on, why this is so important and what are the kinds of things that we hope to be able to do with this data as it begins to amass in LUCID.
Yes, thanks, Andy. We are in an age of big data. We're getting lots of data from tumors now that we couldn't even get 5 or 6 years ago. And my concern is that this data is sitting in silos. They're sitting in silos at specific cancer centers. They're sitting in silos at pharmaceutical companies. But imagine if we were able to take data from a handful of patients or even 50 patients and now have all of that data put together in one database that would be of 500 patients, of 1,000 patients, treated with different types of therapies. Imagine what we could possibly learn about this disease and how much more quickly we can learn. So we're learning a lot from other databases. In fact, ever since the sequencing of the human genome, we have made amazing progress. That's why we're in this biomedical revolution right now. We're learning so rapidly. And the concept here with Lucid that is going to be so powerful is that Lustgarten is funding many science and patient clinical trials. And this is through a new initiative that they have, which is called the Clinical Accelerator Initiative, where they fund small clinical trials at a number of different cancer centers. But the concept is to do a deep dive in the tissue, to be able to do genetic profiling, epigenetic profiling, and proteomic profiling of the tumor tissue as well as the peripheral blood to get a better sense of how the different treatments are affecting the cancer. And sometimes we're going to see sensitivity, so the cancer is going to shrink. A lot of times we're still seeing resistance. But if we can figure out what those mechanisms of resistance are, we can have a better chance of targeting with new drugs. And so what we're planning to do with Lucid is the clinical trials that are being funded, we are asking for permission from the different centers and asking them to ask their patients for permission to put this data into these databases that will be available to investigators who are studying pancreatic cancer to learn how the tumors are changing and to be able to more rapidly develop new drugs. I think this is going to be the biggest advance in clinical pancreatic cancer research that Lustgarten will be funding over the next 5 to 10 years. I really believe that it's the data that's going to drive the development of new therapies, and it's going to help every patient with pancreatic cancer in the future. So I am so excited about this initiative, and I am so grateful that the Lustgarten Foundation is willing to take it on, but it is costly, and we need to make sure we can get the funding to continue to support this initiative.
Thanks, Dr. Jaffe. And I'm also extremely excited to see this, and I can't wait until a year from now, a couple of years from now, where we're back talking again and talking about all of the new ideas, new trials that have spun out of analysis that came from this database, new treatments that are being tested, and new ways to think about and care for our patients. I think it's truly something that's going to be impactful for years to come. So among the very exciting advances that have happened over the 2023, one of the ones that's important is that we're not slowing down. We're continuing to move forward. And Lustgarten had another banner year for funding new research. In fact, we funded $23 million in new research grants in 2023. That represents 17 new grants across 13 institutions across the whole spectrum of research that we support, including continued support for our the crown jewel of our grants program, the LABS program, where a lot of innovative results are coming out of these top cancer research institutes that we support in a sustainable and a large way. It also involves support to young investigators that are up and coming with new ideas, including expansion of our Ruth Bader Ginsburg and John Lewis career development programs that support women in pancreatic cancer research and folks from underrepresented groups in pancreatic cancer research. Those with brightest minds with the best ideas, to support them early in their career to make sure that they can stay dedicated to pancreatic cancer research. It also includes expansion of our LEAD program, a program that's really intended to make sure that when we do clinical trials, that we're involving the whole of the patient population that we want to treat in the end of the day, and that access to clinical trials is more equitable than it is today. This also involves expansion of our Clinical Accelerator Initiative program and funding of 3 new trials as part of that program. So all of that adds up to that we will continue to have innovations, we will continue to have breakthroughs in the years to come, and that keeping that momentum going and moving forward. But in order to maintain that, relies on continued support from our community, from the folks that have supported us to date to keep driving forward.
I continue to be inspired every time I hear these remarkable scientists, and hope you are as well. None of this work could happen without your support. Please consider the Lustgarten Foundation in your year-end giving plans. Because, as you have just heard, we are making real progress in achieving our singular mission, transforming pancreatic cancer into a curable disease. Happy holidays to you all.